All right. So in the previous class, I told you that cell division is not complete without cytokinesis, because what has actually happened, it's simply that the cell has duplicated its nucleus, but it's still one cell. So until cytokinesis happens, the cell has not become two. And if you remember, I used an example that it's going to be like a cell, like an egg with two yolks. So until cytokinesis is completed, that is when the cell actually splits. Mitosis is not splitting of the cell. Mitosis is duplication of the chromosome of the nucleus. So we call it nuclear multiplication. All right, so I'm going to go straight to talk about the phases of the cycle itself. Now, so when you look at this now, for instance, this shows you the cell cycle. Now, there's difference between cell cycle and the proper cell division. Cell cycle tells you the life cycle of a cell. So the cell cycle include interphase, mitosis, and then cytokinesis. So you see, there's interphase, which is the majority part of the stage of the process, mitosis and cytokinesis. Now that was why I told you that to think that nothing happened in interphase is to blackmail the stage, because think about it. You study to take an exam and you study for about 3-4 weeks, and then you went ahead to take an exam of 1 hour, and then somebody came to you and said you have only done the job. Now everything you have been doing is nothing. So does the person want to say that studying for 3 weeks, 4 weeks is nothing? No, because studying for the exam, preparation for the exam, is actually the exam itself. That's what determines what's going to happen on the exam. So interphase is not just a lagging phase. It is a state when the cell is getting ready to divide. All right. So there are 3 stages that characterize interphase. There's growth phase 1. Some textbooks call it growth phase 1 or gap phase 1. So you see here is first gap phase 1, G1. You could see it as growth phase 1, G1. It's the same thing. Then you have the S phase, which is the synthetic phase. This is the stage where the DNA is going to be duplicated. And then you have the second growth phase. Now, then after that, the cell is now going to go into the M phase, which is the mitotic phase itself. That is actually the cell division. All right, so let's go. Now the interphase is divided into 3, like you see here. The cell grows all the 3 phases, but chromosomes are duplicated only in the S phase. So if you look at this, you see G1 says there are metabolic activities going on and growth. So it's at G1 that the cell duplicates its chromosomes, duplicates its organelles, duplicates its protein, and then grows. Then when it gets to S phase, there's going to be DNA synthesis, so that the DNA is duplicated to become 2. Then when it gets to G2, the cell grows more. All right. And then when it gets to after G2, it then goes to the M phase, which is the mitosis itself. We'll be looking at that. Now, ideally, some textbook will tell you there are 4 stages. Some textbook will tell you 5 stages, but it's all the same. Saying there's a pre or real phase is still all telling you the same thing. So don't be confused if you have seen 4 or 5 in any textbook. Now, the whole process is called IPMAT. The whole process is called IPMAT. I'm going to put that on the screen here. So it's called IPMAT. IPMAT. The IPMAT, I is interphase, P is prophase, M is metaphase, A is anaphase, and T is telophase. Those are the phases of the of mitosis or the cell's division. Now, the first thing that happened, so if you look at this now, this shows you, so here, for the 5 stages here, you have prophase, you have in quotes pro-metaphase. Pro-metaphase simply means before metaphase, and I'm going to show it to you as we go. It's nothing complex. Then you have metaphase, you have anaphase and telophase. After that, you're going to have cytokinesis. Now, the this is what the cell looks like at G2. When G2 ends, you have the cell look like this. Now, this is the cell at G2, ready to divide. And that is what it looks like. Now, at this point, you will see that the chromosomes are now obvious, unlike when they were just like granules in chromatin. Now, this is the cell at G2, like this. Now, the first thing you are going to notice after interphase, the difference between this and prophase. In prophase, you will notice that the cells will begin, the chromosome will start to condense. So at this point, you see that they have not condensed as what is here. So the first thing that will begin to happen in prophase is that the chromosome will condense. Why is the chromosome condensing? Like I told you, it is easier to divide what is condensed than to divide what is scattered. If I wanted to divide cereal, let's say cereal into two now. Let's say you have a cereal and I want us to divide into two. If you scatter it all over the house and then we want to divide it, it's difficult to divide in that manner. But if you gather it in a bowl, then it is difficult to split it into two, to divide it into two. So something that is gathered, condensed, is easier to divide than something that is scattered. So for the chromosomes to be able to divide, which is what we want to do, it is appropriate to gather it together first to condense it. So the first thing that happens in prophase is that the chromosome will condense. So you see the chromosome condensing. Second thing that is going to happen in prophase as well is that the nuclear envelope will start to break down. Now, why would this is the nuclear envelope in interphase, but in prophase, it will start to break down. Why do you think the nuclear envelope is going to break down? The nuclear envelope is going to break so that the chromosome can travel out like it's traveling out here. If it is stuck in the nuclear envelope, then it will not be able to travel and spread out. So the purpose of the breaking is so that it can spread out to different regions and migrate to different areas so that two cells can be made. That's the purpose of the nuclear envelope breaking. The third thing that is going to happen in prophase is that spindle fiber will begin to form. So we call this spindle fiber. Spindle fiber will begin to form. Why is spindle fiber forming? Spindle fiber is forming so that they can hold the chromosome in place. All right. So those are the few things that happen in prophase. I mean, you are going to watch the recording again, so I don't need to repeat it. The next thing is actually the metaphase. So this is the metaphase. The thing that characterizes, I'm going to go back to metaphase, don't worry. What characterizes metaphase is that the chromosomes are all arranged at the equator or metaphase plate or at the middle. It's as simple as that. You can say the chromosomes are arranged at the middle, it's arranged at the equator, or it's arranged at the metaphase plate. Now, the difference between this and prometaphase is that the chromosome has started to arrange. They have started to be connected to spindle fiber. So you see some chromosomes are connected to spindle fiber, but they are not at the middle yet. So the journey between prophase and metaphase, in between that is where we now come prometaphase, meaning that before metaphase. It's not prophase and metaphase. No, it simply means pro-metaphase, before metaphase. It's in between prophase and metaphase. It's before metaphase stage. So that's where the word pro-metaphase came from. Now, what happened in metaphase? At metaphase, the chromosomes are arranged at the middle. You see the chromosomes are connected to the spindle fiber, and there are some spindle fiber that we call kinetochore spindle fiber or kinetochore microtubule. If you look at this here, you will see kinetochore microtubule here. What that means is that they are connected to the kinetochore. What is the kinetochore? Kinetochore is a protein that holds the chromosome together. It's called kinetochore. It's a protein. So you see, this we call it non-kinetochore microtubule because they are not attached to any kinetochore. They are not attached to the protein. But these are attached to the protein. So we call it kinetochore microtubule. All right. Then, so at the metaphase, the chromosome are lined at the middle. That's the major thing that characterizes metaphase. Now, prophase, what happened? At the prophase, the only thing that happened is this. Look at it. With this here, we call it centrosome. This is what is here, centrosome. Now, so this is a centrosome. All right. We call it individually, we call it centriose. As a pair, we call it centrosome. So this centrosome, this is another centrosome. The centrosomes are the ones that send out the spindle fibers. So when it became time, the centrosome says, I need my spindle fiber back to me. Can you get me my spindle fiber? Then the centrosome have a way of pulling back its own spindle fiber. So what happened is that each centrosome at each side begins to pull back their their spindle fiber. As they are pulling back their spindle fiber, don't forget the chromosomes are attached to the spindle fiber at the kinetochore, the point where they join. So as each centrosome is pulling back its spindle fiber, what's going to happen? The chromosome will break into two because both of them are pulling back their spindle fiber. So as they are retracting their spindle fiber, each spindle fiber pull out each strand of the sister chromatid. So sister chromatid will separate and go to each pole. Now take note, what is separating? Sister chromatids. So sister chromatids separate and follow each spindle fiber to the respective side. And that is what happened here. Begin to happen there at anaphase. Anaphase, you see, they are now separating. We call this chromatid. Now, what is separating? It's not chromosome that is going to different side, it's chromatid going to different side. Don't forget, two sister chromatids make a chromosome. At the end of the day, in telophase, cleavage furrow begins to form. So this actually begins to form in telophase. It begins to form there already. So it's forming. Then the last stage, cytokinesis, is going to come when this will close up and split into two. In animal cell, we say cleavage furrow forms. In plant cell, we say cell plate forms. So if this were to be happening in onions, for instance, now cell plate is what we form, not cleavage furrow. All right. So this is all about mitosis. I've actually explained it. Any other thing I'm going to be doing now is just to flesh it up, to just show you how it goes. But I've actually explained everything now with this. All right. So I've talked about this. I've shown you this already. It's just the same image. So a closer look to the mitotic spindle fiber now. The mitotic spindle is a structure made up of microtubule that controls chromosome movement. You see that? It controls chromosome movement. That's why it can split the chromosome into two, because it's the one that put them in place in the first place. The reason they were able to stay at the middle in metaphase is simply because the spindle fiber held them in position. Now, the reason they also broke apart in anaphase, another way you can call anaphase is separation phase. The reason they could stay together, stay apart in anaphase, is because the spindle fiber also pulled them apart. So the spindle fiber is what that determines the fate of the chromosome. In animal cell, assembly of spindle microtubules begin in the centrosome, a type of microtubule organizing center. The centrosome replicates during interphase as well, forming two centrosomes that migrate to opposite side of the cell during prophase and prometaphase. So you see that now by the end of prometaphase, the first two centrosomes are the opposite of the cell already, ready to release their spindle fiber so that they can go ahead, arrange them at the middle, and then split them together. An asta, which extend from each centrosome, I will show it to you if you didn't notice it before anyway. The spindle include the centrosomes, the my spindle microtubules and the hasters. Now, kinetochore is a protein complex associated with the centromere. I told you centromere is the location where the two sister chromatids are connected, and the protein that is in that region that hold them together is called a centromere. All right. Now, during prometaphyse, some spindles microtubule, kinetochore microtubule attached to the kinetochore. I've told you that already, right? That is kinetochore microtubule now because it's attached to the kinetochore. Others like this now, it is non-kinetochore protein. I mean spindle fiber because it's not connected to any kinetochore, right? Now, at metaphase, the chromosomes are lined up at the metaphase plate and imaginary line midway between the two spindle fibers. So we call it metaphase plate, we call it equator, we call it middle line. Now, in inaphase, what's going to happen? The coenzyme are cleaved by an enzyme called separase. The sister chromatids will now separate. You see that? They will separate because the coenzyme that join them together have been removed. They will now separate and then the tube will move them separately. The microtubule will now shorten. So you see, it's like the microtubule is retracting. It's shortening, and by depolymerizing simply means breaking into pieces. It's breaking into smaller components. They are kinetochore enzymes. Hence. So the result of a clever experiment suggests the motor protein kinetochore work the chromosome along the microtubules during anaphase. The depolymerization of the microtubules at the kinetochore ends occur after the motor protein have passed. All right, okay. So kinetochore microtubule from opposite pole overlap and push against each other, elongating the cells. All right, this is actually what leads to the cell to now become longer. So they keep pushing against each other. At the end of the anaphase, duplicate group of the chromosome have arrived at the pole at the ends now. So at the end of anaphase, this is what you're going to have. All right, it's more like this. This one side, this another side as well. Now, the next thing that will begin to happen will be cytokinesis is going to begin. Now, cytokinesis actually begin at anaphase or at telophase. Usually it begins at anaphase, but it's not completed until after telophase. That's why we say cytokinesis happens there. Is the end of telophase that cytokinesis will be completed, but cytokinesis is already in place. All right. So in animal cell, I told you cleavage furrow will be formed. In plant cell, cell plate is going to be formed instead of cleavage furrow, doing the same job, right? All right. I've told you about this. This is prophase, prometaphase, metaphase, anaphase, telophase. So we actually call it PMAT, P-M-A-T, not regarding this as a real stage. But it's okay to have it there. So this is what happened in animal cells. Cleavage furrow happened, the cells split into two. In plant cells, cell plate happens and the cell split. So the cell plate that in animal cell, cleavage furrow in plant cell. In a cell plate in plant cell, I'm sorry, cell plate in plant cell, cleavage furrow in animal cell. All right, so if you're looking at this under a microscope now, you see this is what you see in plant cell when you are looking at it under the microscope. And you can tell, looking at this, of course, let me use my highlighter now, or annotation rather. Looking at this, you can tell that this here is anaphase, right? This can tell that this is the anaphase that is already fully formed. This is anaphase that is still in process. All right. At this point, you could feel like this is more like anaphase extending to telophase already, right? But not definite yet. But at this point, you see the chromosomes are the middle here, so it's metaphase. At this point, they are already starting to separate. And of course, you can see key thing that you should use if I am asking you. Now, this may not be a good one to consider when I'm asking you to choose something, if I have to do that. But the key thing to check is when you see the spindle fiber, you see this is the spindle fiber here. Those are the spindle fiber. When you see the spindle fiber and then the chromosome, then you see them separating already. So that's anaphase. All right. When you see these ones, serious ones here, these are the interphase. All right. When they now start to condense, you see, at this point, the chromosome is not something you can separate because they are just all together. They are uncondensed. But at this point, you see, they are beginning to condense. This will be prophase. All right. So those are the stages of those are the stages there. All right. So this what we just looked at. These are prophase, prometaphase, metaphase, anaphase. You see, now key thing. I know sometimes it may look a little bit confusing, but you need to get the differences clearly. So if you look at this is prophase, of course, nothing is happening here. The cells are becoming condensed, um, and now they are becoming condensed and visible. Ideally, if it's that you are showing it, spindle fiber will start to be formed here. But here, this is prometaphase. The chromosomes are now attached to spindle fiber. They are ready to migrate, right? Now, here is metaphase because you see chromosome arranged at the middle. All right. Now, at this point, you see that this chromosome that were arranged has started to spread out. So you see that those are different. They are beginning to separate now. This is anaphase. Telophase is when you see them at the opposite side. Then cytokinesis will be completed. Cell plate formation. All right. So prokaryotic reproduce by a type of cell division which we call binary fusion. Binary fission. Fission means to split into two. So in binary fission, this chromosome replicate at the origin of replication. This is what we call origin of replication here. The plasma membrane pinches inward, dividing the cell into two. So this is what you see in prokaryotic cell. We call it binary fission. Fission is different from fusion. We do fusion, sexual reproduction, fuse together. Now, because prokaryotic evolved from eukaryotes, before eukaryotes rather, mitosis probably evolved from binary fission. Certain unicellular eukaryotes exhibit the types of cell division that seem intermediate between the binary fission and mitosis that talk about. So it's okay to think that that is where mitosis originate from. Let's go. I'll just touch base on. Okay. Now, the key things I would like you to note here is that cell cycle or cell division is regulated. And the regulation is such that I'll go back to this image to show you this. Okay, all right. So, I told you, I'll come back to what I skipped. I told you that, all right, I told you that there is in G1, in interface is about the most important phase of the cell cycle, right? Because that's the point is getting prepared for the division. And if it's not well prepared, the division cannot happen. So there are stages in the cell cycle, there are points in the cell cycle that helps to track the system to be sure that what the system is doing is what it should do. So we have different checkpoints. So between G1 and S phase, we have G1 checkpoint. G1 checkpoint is going to majorly check to see if the cell has duplicated its organelles well. It's going to check if the cells has duplicated its proteins well, and then if everything is well, the G1 checkpoint says, okay, go. The cells can now go. Now, if the cell notices that something is wrong, then it can send it to G0. We call it G0, G0. That's the point of killing the cell, apoptosis. So if the checkpoint feels like, oh, this cell is not growing well, it has not met the conditions that should be met at G1, then you can say, okay, this cell cannot fly, then G0. Now, but if everything is okay, it will say, oh, now continue to S phase. Then S phase is going to synthesize DNA. Then that process continues. and then the G2 goes, and then it comes to want to start the M phase. Now, before the M phase is mitotic phase, right? It's going to check again. We call it G2 checkpoint. It's going to check at G2 and say, okay, did this process go well? Has the DNA been properly synthesized? Has the cell properly grown and ready for division? If it is not ready, send it back. If it is ready, then it proceeds to the M phase. Then there is also a checkpoint at the M phase, which we call M checkpoint. Let's go. So there are regulators. Excuse me, please. So there are regulators for the cell cycle. As a matter of fact, cell cycle is tightly regulated. What we actually call cancer today is simply unregulated or uncontrolled cell division. So when cell division begins to control without having any checkpoint, without having any check, that is what we call abnormal growth. It is the abnormal growth that can be tumor, which can be benign or malignant. If it is malignant, we say it's cancerous, it's going to spread. If it is benign, it is localized in an area. It's just going to grow and it can be handled because it's not going to spread. All right. So the frequency of cell division varies with the type of cell. These differences result from regulation at the molecular level. Cancer cells manage to escape the usual control of the cell cycle. All right. So if you look at this G1 phase, this S phase, this G2, and this is mitosis. Each of them have cycling. They have different molecules at different stages that are responsible for the regulations. Let's go. Now, so the cell cycle control system, we're going to start looking at that. The cell cycle appears to be driven by specific signaling proteins and molecules present in the cytoplasm. Some evidence for this comes from experiments in which cultured mammalian cells at different phases of cell cycle were fused to form one cell with two nuclei. I'll talk about that. Signal in the cytoplasm of the fused cells cause both nuclei to enter the phase of the, don't worry. No, I'm not talking about the experiment. All right. So this is a checkpoint. So we call it cell cycle checkpoint. Now, when you look at, okay, so there are two types of regulatory proteins actually, and that's why we talked about cycling in the first place. So we call cyclins. The regulatory proteins are called cyclins or cyclin-dependent kinases, CDK. Now, for some of you that read journals about cancer, you'll have seen CDKs very well or cyclin. Now, we call CDK cyclin-dependent kinases. Cyclins, they are named based on the way they fluctuate. Cyclins, like they are in cycles. All right, that's where cycling came from. So you see they are at different fluctuations at different level. Then the activity of the CDK rises and falls with changes in concentration of the cyclin as well. So cyclin fluctuates. The fluctuation of cyclins also affects CDK. For any CDKs to be active, they have to be attached to a cyclin. Now, if they are not attached to cyclin, then they are not active. All right. So if you look at this now, this CDK, it is inactive because there's no cyclin there. When cyclin binds to it, it becomes active. All right. Now, so that's the way the cycle works. All right. So MPF simply means maturation promoting factor that help to promote factor maturation, and it's simply the coupling of CDK and cyclin. Like I told you, CDK is only active when it's bound to cyclin. The CDK cyclin complex that triggers cell passage has the G2 checkpoint into M phase, right? So after the G2, they come to certain place. The peak of MPF activity corresponds to the peak of cyclin concentration, of course. MPF adds both as kinase. What are kinases? Kinases are encode proteins. They are enzyme, of course, ASE, that help to they actually, the best word to use is that they help to phosphorylate. Of course, we've done cellular respiration, for instance, I could talk like in those terms. So kinases, they are proteins that help to phosphorylate another protein so that it will be active. So when there's a need for ATP now for a cell, for a protein to do a job, kinases are the one that will give ATP, give the terminal phosphate of that ATP to the substance so that it can be activated, energized. So they are the one that phosphorylate, which is in turn giving energy, right? So MPF act as kinases, so that's what they do. They act as kinases and indirectly through activating other kinases. You see that? So they act as kinases, they also indirectly they activate other kinases by giving them energy. All right, let's go. It's still the same slide. All right, so there are three important checkpoints like we talked about. The checkpoint are the G1 checkpoint, G2 checkpoint, and the hem phase checkpoint. So this is the hem phase checkpoint. This is the G1 checkpoint that can, at this point, like I said, okay, let me look at this from here. So without go ahead and signal, the cell exits the cycle and goes into G0. So that's what happened here. So if the cell is not authorized to go ahead, then it can go to G0, which is what we call apoptosis, if it cannot pass through the G1 cell cycle. Now, then if there's a go ahead, the cell goes and then continue the process. All right. Now, for many cells, the G1 checkpoint seems to be most important. If a cell receives a go ahead at the G1 checkpoint, then the cell will likely complete the other stages. However, if the cell does not receive go ahead from the G1, then it's likely going to be sent to G0, G0, which is apoptosis, of course. All right, the same thing we looked at already. So we've talked about mitosis, metaphase. I will not talk about that again. Let me see if there's still anything I want to touch before we go. Yeah, just read those. All right, so cancer cells do not eat to normal signals that regulate normal cell activities, and that is what we call uncontrolled cell division. So you see a tumor growing in the palace of a cell of a patient. Ideally, that should not happen, except that there is growth that is not controlled. So they do not stop dividing when growth factors are depleted. They continue to grow because now they don't have anything regulating their growth. They are people without control. I mean, they are cells without control. So we call that cancer cells. So cancer cells do not need growth factor to grow and divide. They may make their own growth factor. They override the system. They may convey a growth factor signal without the presence of a growth factor. They may have abnormal cell cycle control system by themselves that is different from the regular body control system. And so they go ahead and overdo their growth. All right. Now, cells that some cells, so this is it. There is the word we call transformation simply means a cell is taking up a new trait that it doesn't have before. So let's say I just decide to learn Spanish today better than the poquito that I speak. If I decide to learn that more and it becomes part of me, I have been transformed. So transformation simply means when a cell picks up or gets a new strain of DNA segment into its DNA, and that alter some stages or some processes in the cell. That's what we call transformation. So cells that acquire the property they acquire the ability to divide indefinitely. We say that the cell has been transformed because a non-transformed cell, which is a normal cell, will not have such traits. So a cell that keeps dividing without control will likely be a transformed cell. Cancer cells that are not eliminated by the immune system form tumor on the long run, and the masses of the abnormal cell within otherwise normal tissue. If abnormal cells remain only at the original site, we say that's benign. If it's spreading, we say that is malignant. All right, that's malignant. So the property of malignant cells or growth tumor that makes them to spread, we call it metastasis. So metastasis is to spread. All right, is to spread from one place to another, whereas tumor or benign will be localized in a location. All right. So localized tumor may be treated with high energy radiation, which damages the DNA in the cancer cell. The majority of cancer cells have lost the ability to repair DNA damage again at that point. All right. So, all right. So metastasis is when when there is tumor and the tumor continue to increase, then continue to spread from where it is. And so that's why they say there is a cancer of the breast, but now the cell is no more localized in the breast, it has spread around throughout the body. That's what we call malignant. The ability of that cancerous cells to spread out of its hidden places, to spread out of where it's localized and circulate through the entire body. That is what we call malignant. But if it is just benign, it's going to be in a location just like what you see here. This is benign. This may be benign if it's just localized here. And all that needs to be done here will be a surgical procedure, then check what is the cause and undo what is the cause, and then that may not happen again. But if it is benign, if it is malignant, and it is not taken care of on time, perhaps at stage 1, then the risk of spreading out becomes a problem. And that's why cancer that are detected at the early stage have high it has easier management than cancer that are discovered at like later stages when it is already spreading out. All right. So malignant tumors invade the surrounding tissues and can undergo what we call metastasis, which is spreading. The spread of the cancer cell to other part of the body where they may form additional tumors. That's actually the problem with cancer cell. Localized tumors may be treated with high energy radiations, which will damage the DNA. The majority of the cancer cell have lost its ability to repair DNA at that point again. So that is what we have. All right. Lastly, metastatic cells tumors are treated with chemotherapy therapeutic drugs that target the cell cycle. All right, so you see, target the cell cycle as though to hinder the replication that has been uncontrolled. It's just like a vehicle lost its brake. The vehicle brake got bad, and then it's going to lose control and keep moving. Right, so anything you want to do to that vehicle is to see anything to stop it. All right, so that's what chemotherapy medications are for. The side effects of chemotherapy are due to the effect of drugs on the normal cells. So don't forget, and that's the challenge with viral infection, right? When you have viral infection, the medication you take for viral infection is also going to affect your regular cells because virus is eukaryotic, just like your body cells are eukaryotic as well. It's easier to kill bacterial cells because bacterial cells are prokaryotic, whereas your body cells are eukaryotic like viral cells. And that's why you don't take antiviral infection, right? So the danger of the challenge that chemotherapy also have is that it's going to affect it's not just targeting the cancer cells. Of course, it's going to have impact on all the body cells. That's why you notice that people get weakened after taking chemotherapy because it's going to weigh down the whole body. It's also going to hit on the uh, normal cells in the body. Researchers are producing a flood of information about cell signaling pathway and their relationship to cancer, coupled with new molecular techniques that can be better treatment that will be attacking the tumor cells itself and not all other cells. All right, that's where we are. If that could be success, if that is successful, that would be a great one. If there's a way to target the cancer cells alone with chemotherapy and not generally exposing the whole body cells to that. All right.